Effects of artificial sweeteners on biofilm formation in model gut and oral bacteria. Artificial sweeteners are zero-calorie sugar substitutes found in various products, including gum, candies, and beverages. They gain popularity due to their ability to help manage weight and replace sugar, which is associated with bacterial growth and health issues such as dental caries and gut biofilm formation. Biofilms are bacterial communities encasing protective layers, making them resistant to antibiotics and contributing to many diseases. Biofilms have been linked to causing dental diseases and dental caries, and they have also been linked to causing gut diseases like irritable bowel syndrome, periodontitis, and even gastric cancer. Despite their widespread use, research on the effects of artificial sweeteners like aspartame and sucralose on biofilm formation remains inconsistent, creating a significant gap in understanding their impact. Studied by Manjula Weerasekera, the data presented information on how saccharin influenced biofilm formation in specific bacterial species. The graphs show that saccharin significantly inhibited biofilm formation when compared to the groups with sucrose and glucose. In a study by Aparna Shil, it expanded the scope to compare multiple artificial sweeteners. The results showed differential effects of the sweeteners aspartame, sucralose, and saccharin. The artificial sweeteners caused an increase in biofilm formation when compared to the control group. There is a significant gap in research regarding the effects of artificial sweeteners on biofilm formation, as existing studies present conflicting views, with some reporting inhibitory effects while others indicate no impact or enhancement of biofilm formation. The study aimed to investigate the effects of artificial sweeteners, specifically aspartame and sucralose, on the formation of bacterial biofilms. The research question posed was, do aspartame and sucralose affect the bacterial biofilm formation? It was hypothesized that exposing E. coli K12 and Streptococcus mutans to these sweeteners would result in increased biofilm formation compared to the control. Materials needed are a biofilm assay kit, flasks, E. coli K12, a Streptococcus mutans, and a microplate reader. To test the hypothesis, E. coli K12 and Streptococcus mutans were grown in media containing different concentrations of aspartame and sucralose. The biofilm assay kit was used to stain biofilms with crystal violet solution, and biofilm formation was quantified by measuring absorbance at 630 nanometers with a microplate reader. Results showed that both aspartame and sucralose inhibited biofilm formation in E. coli. For Streptococcus mutans, the inhibition was even more pronounced, reinforcing the potential role of artificial sweeteners in reducing biofilm-associated issues. Aspartame exhibited a stronger inhibitory effect compared to sucralose for both tests. ANOVA analysis confirmed significant differences in biofilm formation across treatments. The study found that artificial sweeteners reduced biofilm formation, a result that was unexpected and contradicted the initial hypothesis. Aspartame demonstrated a greater inhibitory effect than sucralose. This suggests that artificial sweeteners can interfere with bacterial adhesion and biofilm matrix development. Additionally, the experiment provided valuable learning experiences such as mastering anaerobic bacterial culture techniques and understanding biofilm resistance mechanisms. Additionally, the ANOVA test result and a p-value of less than 0.05 showed that the results were statistically significant. It was unexpected that aspartame had the greatest inhibition of biofilm formation when compared to all of the sweetener types. Findings demonstrated that artificial sweeteners, particularly aspartame and sucralose, inhibit biofilm formation rather than promoting it, as initially hypothesized. This discovery has significant implications for reducing biofilm-related health problems, such as dental caries and gut biofilm disorders. The inhibitory trends observed in the graphs highlight the potential for artificial sweeteners to be used in therapeutic strategies, offering a promising avenue for future research and practical applications in healthcare. Assistance throughout the project was received from Mr. Stafford and Miss Young, who offered guidance and advice throughout the process. The experimental design was strong because it was an efficient biofilm formation assay. The 96-plate lid format made it easy to equally wash and stain each group. It was cost-effective, easy to use, and ideal for testing various independent variables and established biofilm quantification methods. However, several limitations were identified. Equipment constraints such as the use of a 630 nanometer microplate filter instead of the recommended 590 nanometer filter may have affected precision. Additionally, the study focused on single-species biofilms, limiting its applicability to more complex, mixed-species communities commonly found in natural environments. Time constraints and variability in bacterial preparation were also noted as potential sources of error. Future research should explore the effects of natural versus artificial sweeteners on biofilm formation. Studies could focus on mixed-species biofilms to better replicate real-world conditions and investigate the long-term effects of sweeteners on biofilm-associated diseases. Furthermore, testing the genotoxicity of artificial sweeteners and expanding the variety of tested bacteria and sweeteners would provide a more comprehensive understanding of their impact. These steps could help clarify whether artificial sweeteners could serve as tools for managing biofilm-related health issues.